Welcome back, everyone. Next, we have Regen BioPharma Inc. It trades on the OTC Pink sheets under the symbol RGBP. It's a biotech company focused on developing innovative treatments using autologous cell therapies, RNA and DNA-based immunotherapy, and small molecules in the immune-oncology and autoimmune disease space. Happy to welcome back David Koos, President and CEO, and Harry Lander, Senior Scientific Consultant. Welcome, gentlemen. How are you doing today?
Well, thank you very much. That's great. I'm excited about some developments we've had that I think should be really interesting to our shareholders.
Wonderful. The floor is yours, and you call me back when you are ready for questions.
Okay, perfect. So this morning, I think we're going to cover recent developments in our science regarding the CAR T cells. We're also going to cover equity line financing and what that all means. And then we should have a little bit more on some of the other business aspects. We're going to get into a presentation, brief presentation by Harry, and then we'll go into some questions and answers for the various aspects of what's happening with our CAR T program. We'll transition from that to the equity line. And then at the very end, we expect to answer questions from the audience so that we don't let anybody walk away feeling like their questions weren't answered or somehow addressed. The goal is to be transparent. The goal is to have our shareholders comfortable with the direction that we're taking and where the company's going with its science. So with that, I'm going to start out with Harry and let him jump in the middle with both feet and talk about this study that we just completed and what that means. Harry, take it away.
Sure. Thank you, David. Maybe before I jump into the details of that study, maybe I can give a little overview of sort of the scientific approach that the company has been taking on NR2F6. So NR2F6 is a gene that the company has been studying for many years. The reason that we have been focusing and targeting that gene is because it has been clearly shown to be an immune checkpoint. Immune checkpoints are proteins that prevent T cells from becoming too activated. So for those, many of you probably know this story, some of you don't. And for those who don't, you think of T cells as an army. Sometimes you want it turned on, sometimes you want it turned off. And if you're the immune system and you're attacking cancer or you're attacking a virus or bacteria, you want to be on it, but when you've done your job, you need to be turned off. And immune checkpoints play an important role in that. But it turns out that tumor cells have ways of avoiding the immune checkpoint and keeping it turned on, so preventing the T cells from activating and killing. So, and T cell immune checkpoint inhibitors are drugs that are on the market, as many of you know, billion-dollar market, because they are very good at releasing inhibiting the checkpoint. So if you inhibit the inhibitor, you get activation of these T cells. And so they're used in many, many kinds of cancers, very effective. NR2F6 is also a checkpoint. And we've developed several small molecule drugs that inhibit the NR2F6. We have a patent around that. We've developed drugs that activate NR2F6, which has likely the opposite effect, right? And so that could potentially suppress the immune system, which can be very useful. We have a patent on that. And then we have genetic approaches to do both of those things, inhibit and activate NR2F6. And we have patents filed around that. So that's why we focus on the NR2F6 program a great deal. And we've had a lot of sort of efforts on that. So this program that David is referring to, which we call the DuraCAR program, the idea is to genetically inhibit NR2F6 in the context of CAR T cells. So CAR T cells are T cells that are, there's currently drugs on the market called CAR T cells, where one takes the T cells from a patient who has a cancer, a treatable cancer, and one of the proteins on the T cells are modified. It's called the chimeric antigen receptor, CAR. These CARs are modified so that they have on the outside, because they stick across the membrane of the T cell. So on the outside of the CAR, you have you put something on it that targets it towards the tumor you want to kill, most commonly B-cell tumors. And so that brings the army, it brings those T cells directly to the point where they need to recognize this as a cancer cell and then do their killing. And so that's very effective for certain cancers, mostly liquid tumors. It doesn't have real effect yet on solid tumors, and we're going to get to that. So what we wanted to do was create these type of CAR T cells, but in addition to just targeting them towards a specific cancer, on the inside part of the protein, the part that's inside the cell, we put a what's called an siRNA, something that inhibits or should inhibit the expression of NR2F6. So the idea is that now we have these highly activated T cells that they're immune checkpoint inhibited by inhibiting the NR2F6, and now we can activate these T cells and get tremendous killing. That is the approach. So it's a bit of a complicated process because you have to create these bifunctional. These CARs have two functions. They have the outside part and the inside part. So we designed the experiments. We hired a contract research organization to perform those experiments. And it took some time to do. As I said, it's a bit complicated. But they were able to engineer these CAR T cells and then test whether NR2F6 was indeed inhibited inside the cell. Well, they found something unexpected. They found the opposite, that the NR2F6 was actually the expression of that of the mRNA was activated. That didn't make a lot of sense to me mechanistically as a scientist, but the data are the data. But we wanted to double check. So we had them try 5 different types of siRNA. So basically 5 different types of that CAR T, and every single one had the NR2F6 highly expressed. So that was sort of unexpected, but it seemed true. But to me, it was so unexpected that we elected to hire a second contract research organization, completely independent of the first one, in fact, in a different country, and have them repeat the experiment. They found the exact same thing. So the data are true. The hypothesis was wrong, and the data are true, which is terrific because now we have potentially a T cell that has high expression of NR2F6, a CAR T cell, and one would expect that these CAR T cells would be immune suppressive. And this is a very large potential market because these T regulatory cells or these immune suppressive T cells could, in theory, prevent autoimmune disorders or stop autoimmune disorders. So, for example, in type 1 diabetes, you have the immune system, for some reason that's not known, recognizes your own beta cells as foreign and destroys them. And so you have diabetes, you can't produce insulin. And that's a terrible disease, type 1 diabetes. But there are many autoimmune disorders. It's a huge market, much bigger than cancer. And so we have, early stage, but a potential to develop a cell therapy targeting autoimmune diseases. There's a couple of companies out there that have begun trying to use cell therapies to do this. One was just bought for a lot of money. So we are now preparing a new set of experiments to ask the question or answer the question, do these T cells have immunosuppressive properties? Can they suppress the immune system? At least initially we'll look at sort of, at a high level, can they suppress targeting of normal CAR T cells for killing cancer cells? So that'll take some time to do. Those experiments typically take maybe, they'll take about, let's say 6 weeks, who knows, but approximately 4 to 6 weeks to get those data and analyze them. If we in vitro can see that we are actually suppressing the immune system, then that opens a huge area for us, and we would have to really think hard on exactly which indications to target, what kind of T cells to create, because these are great, but we would probably want to change that outside part that is right now targeting tumors. We would want to focus it, of course, on bring these immunosuppressive cells to areas like in arthritis, lupus, type 1 diabetes. There's many options, colitis, where we could potentially suppress the immune system and have a beneficial effect. So, but one step at a time, and the best thing about this is that we're getting real data that is not, neither here, neither there. There's clear, in our case, activation of NR2F6 at the moment.
Harry, let me jump in with a question for you. So as we go through this and you're talking about NR2F6, why did we choose NR2F6 as opposed to some other indicator?
Yeah, so NR2F6 is poorly studied. When I say poorly studied, the people who have done work on it have done excellent work. There's several excellent publications in the literature around NR2F6, but there are very few studies around NR2F6. So it is prime to be looked at as a druggable target. Now, the NR2F6 gene is a nuclear receptor, and it's one of these what are called orphan nuclear receptors. And these they're called orphan nuclear receptors because no one really knows what naturally binds to them in the human body. And that's one of the reasons it was a big deal for us to find small molecule structures that seem to actually bind to NR2F6. No one else has done that. And we were able to get a patent around that pretty easily. So it's a very interesting molecule. I think it makes a lot of sense scientifically and strategically to go after NR2F6. It's not an easy target, but there are reasons that it's not well studied. So to answer your question, David, we've continued to focus on it because of those reasons. It's a prime target as an immune checkpoint. It is not that there aren't that many groups working on it. As far as I know, there's not a single biotech company focused on it, so it makes a lot of strategic and scientific sense to go after it.
Okay, so now I know in the past we've had the approach of working on the concept of creating small molecule therapies. Now we're in the CAR T, and I'm trying to figure out maybe how those dovetail together, or if they dovetail together.
Right. Well, in some ways they complement each other. The nice thing about the cell therapy approach is that you're going to have a very directed approach. So whether it's cancer, or let's use the type 1 diabetes example, for autoimmunity. If we are able to create immunosuppressive cells that can specifically target the pancreas, you're not targeting the rest of the body. When you give a small molecule, you're targeting the entire body. So side effects, unwanted effect, off-target effects are there. So if you are suppressing the immune system, let's say with a small molecule, like prednisone, like a steroid, you're suppressing the immune system throughout the body. With these types of cells, you're being very specific and able to create very localized treatment. So that's one advantage of the cell therapy. There's no doubt the cell therapy is more expensive by far, but there are plenty of cell therapies that are being used right now that are FDA approved and are covered by insurance. So it's out there. And as time goes on, I really believe this is sort of the decade of cell therapies. You know, there are many companies that are working on all kinds of cell therapies, not just for cancer. And it's really the next evolution after small molecule drugs and the biologics. So, but you know, having the small molecules in hand is a great tool and something we can certainly pursue if we had the funding. And there are many companies that are historically very expert at small molecules, like Pfizer, like Merck, that don't necessarily want to get into the cell therapy space. So having both, I think, is quite complementary.
Now, I'm curious, do we have any notion that potentially cell therapy may work better on autoimmunity and small molecule may work better on cancer? Or is that just me sitting here drinking coffee and thinking about it?
No. That's interesting. So if you're going to inhibit NR2F6 with small molecules, let's say, and therefore activate the immune system, you're inhibiting NR2F6 throughout the body. So would that have an effect outside the immune system in a negative way? We don't know. That's typically what a phase 1 trial would determine. So that's an interesting point. And then on the activation side, if we gave small molecule full body, so there you'd be suppressing the immune system entirely. So I would agree with you. So there's an advantage to using a cell therapy approach for autoimmunity if that pans out, for sure. Because you know, people now with autoimmune disorders, they take steroids, which has tremendous side effects, or anti-TNF alpha antibodies, antibodies that affect. So yeah, I agree with you, David, that using cell therapy towards an autoimmune approach makes more sense than using a small molecule approach.
So the results that you just described on the studies that we just completed, where do you see that leading to? I mean, do we have a new next step that we're looking at, and where are we in the process of selecting a contract research organization that could step forward and be the right one to pick?
Yeah. So of course, you know, we want to get this into animal studies as soon as possible, right? But we can't jump the gun. We have to, you know, do it properly. So I've put out for bid the studies that I described, the next set of experiments in vitro to determine exactly what we have, and so we expect a response shortly. So we'll have a couple of CROs to choose from, and then we'll make the decision. As of course, David will make the decision as a company and select one. But once one is selected and we enter into the contract, the experiment won't take too long. As I said, I think maybe 4 weeks or so for this initial experiment. We'll have some results.
So that's 4 to 6 weeks from the point that we select the CRO and we believe that we're close to having one that we want to use.
Yeah, maximum. Yeah.
Okay. Good. So at this point, when do you think we will attract the attention of big pharma as to what we're doing?
That's the $1 million question, or the $1 billion question that all the biotechs ask. It's for sure it's too early, but if we could get this, you know, to an IND, that will attract attention, and then of course completing a phase 1 trial would attract a lot of attention. So the big boys, as we call them, they're not interested in general at this earliest stage. Perhaps, you know, if we can get through animal studies and show good results there and get a publication out there, then I think we'll have probably some early discussions. But yeah, so it's definitely a little early for that.
Okay, so where we're at right now, if we were to continue with success in the various stages that we have outlined, do you see any larger biotech companies that would be thinking in terms of some sort of a relationship with us? Maybe not an acquisition, but a shared research development, something to that effect. Does that make sense, or are we still?
No, that makes sense, and I think that's definitely in the realm of possibilities. We just need to, because it's so novel, and there was a recent acquisition. I forgot the numbers, but they were big numbers, where Encarta, a small, preclinical company, had some terrific data, and they just entered into a giant deal. I forget these numbers, but we're talking big numbers. So, and it's so new to if we go down the T regulatory pathway, the autoimmune pathway, I think that would be there would be a lot of excitement out there.
Okay, well, that's interesting. I'm sitting here and I'm looking at my watch, and I'm thinking that as much as I could spend another half an hour with you talking about this easily, but I think we probably need to move on to
Sure.
The big question: how do we finance all this? And so I think we've mentioned several times now that we have an equity line in place with a fund that is there to assist us so that we can keep moving the science forward. The thing that we look at in the past, unfortunately, we've used a lot of convertible debt that I had to spend a great deal of time cleaning up when I first came back to the company. The goal with the equity line is we can use it or not use it. It's entirely up to us, the company, as to what amount of stock we need to place and how that impacts the shareholders. I'll give you an idea. We haven't been very aggressive in using it at this point. We look at that as a backstop if needed. I think we've placed maybe a total, since it started, of around 50,000 shares. So it hasn't been all that much. The idea is that we don't want to use it any more than we absolutely have to. I'm committed to seeing that this company gets to where I think it has the potential to go. To this point, I'm still not drawing a paycheck out of the company. So that tells you I believe in what we're doing. And the equity line makes sure that we're not sitting here trying to figure out how to keep the science moving forward. That's the big issue on the table, especially with the developments we've had in this last study that we did in the confirmatory study, is that if you get that kind of information, you don't want to take a pause and say, well, we're taking winter break or something. You want to keep the momentum going. You want to make sure that where we sit, we're able to keep the momentum going. And that at the end of the day, we believe we have something very special. So our goal is really to keep things moving along. And as opportunities come up where, for example, we get some more of the steps out of the way to make us look attractive in a partnering relationship to develop the research, that would not be dilutive to shareholders, because it would most likely be we have the science, they have the cash, and together we're developing this technology. We have to get to that point, which I don't view as being too terribly far down the road, But we do have to get to that point to make sure that we can attract the kind of capital infusion that we can be ready to move into the IND phase and move into clinical trials, phase 1, phase 2. But at that point, I feel extremely comfortable that we will have enough data and enough interesting insight as to how NR2F6 works using our CAR-T therapy. Then it's a matter of moving forward. We're going to have to select programs that make sense for us to move along with, indications that we want to experiment with and see where we can go, how far this takes us, and hopefully we have a very positive impact on human health. So that's the goal in the whole thing. It looks like we don't have that much time left, so I'm going to go ahead and start taking questions from the audience. Here again, feeling like we're in a position that we could keep going for a long time. All right, so Anna, what do you have coming out of the participants in the way of questions?
We should just have the Regen conference just for you guys. We've got so many questions for you. Next time, let's schedule that. All right, we do have a few minutes. Let's see how many we can get in. Carl Roca says, I think you mentioned a company like Merck would not want to get into the cell therapy space. So do you think that since you are, it increases your value to the majors?
Gary, do you want to handle that?
Yeah, sure. Potentially, there are certainly a bunch of big pharmas that are involved in cell therapy, Gilead, AstraZeneca. So, but there are some big pharmas that just really don't want to play there. Their historical strengths are in small molecules or biologics. So, but yes, I agree with you that the playing field is smaller, and therefore perhaps our value will potentially be larger.
And Sean Tyler asks, what multiple should RGBP be trading at compared to RGBP?
I've been perplexed at that because I think RGBP, the Series A preferred, there's only about, I want to say, 195,000 shares in the system. And that's just off the top of my head, but I believe that's true. 195,000 shares is nothing. And so I'm wondering why it's so volatile, because there really isn't a lot of stock there. There's no millions of shares. There's a handful of shares out there. So I don't have any way to gauge what a good answer would be at this point, and I apologize for that because it's baffling me.
And Jim McGrath wants to know, when will your Q3 report be released?
Well, right now we're on our annual report. We're working on that, and that should be out. Well, it has to be out by December 31st.
And Dylan Mosley wants to know what trials are currently going on and what data points are next to come in.
Well, I think we sort of alluded to that in Harry's presentation. We've wrapped up the study, the confirmatory study, and now we move on to the next phase. And I don't know, Harry, how would you categorize the phase that we're going into?
It's still in vitro, so we're still testing things out in the test tubes, so to speak, before we put them into animal studies. So it's considered preclinical in vitro studies.
Okay.
And Ryland Cross wants to know, what's your current cash position?
Since we're right in the middle of the 10K being audited, I don't know that I feel comfortable throwing that out on the table until the audit's completed.
Okay, no problem.
Yeah.
We'll send all these questions to you so you can answer on your own time anyway.
Okay.
Andy Costa asks, with regard to the NR2F6, when will we know if this is inhibited or enhanced? And what will either that mean to your timeline?
So if I can take that. So far it looks like it's enhanced by this method that we're using in these CAR T cells. So, but to answer your question more fully, I believe in about a month's time, let's say 4 to 6 weeks, we will know if that increase inside the cell causes those cells to be immunosuppressive.
And Greg Giordano asks, what will the cost be to do animal studies, and how much capital will you need to raise to go into those animal studies?
So the first set of animal studies are typically done in mice, which are not that expensive in these types of studies. To give you a range, it will probably be to do a serious set of experiments to get significant data. It will probably be in the $100,000 range, maybe, plus or minus in that range.
And just last question due to time, maybe it's a nice closing question as well. Mark Yaskouts says, where do you believe you see this company going? Whoever wants to answer that.
Wow. Well, I think the fact that I'm not drawing a salary out of the company, I'm expecting to do well when we've gotten to the point that we have something that Big Pharma is interested in. What that number is, I don't know. But I think it's got to be a lot better than a paycheck.
Okay. All right. Well, thank you, gentlemen, so much for joining us again. We always love your live updates, and we look forward to seeing you again in the near future.
All right. Thank you very much, Anna.
Thanks, everybody.
Okay, everyone. We'll be right back.
Bye.